Now we are going to start learning about renal physiology, the various sections included in that renal physiology like functional anatomy of the kidney, urine formation by the kidneys, renal blood flow, glomerular filtration and their control, along with that tubular function, concentration and dilution of urine, control of extracellular fluid osmolarity and sodium concentration, renal regulation of potassium, calcium, phosphate and magnesium, then acid base regulation, micturition, applied physiology of the renal system, some medical related terms associated with that. So we will be learning all these portions one by one, just with the help of some videos. So first, in this video we are going to learn about the functional anatomy of the kidney, human kidney. So learning objectives include describing the functions of the kidney, describing the structure of the nephron, describe the difference between a cortical and a juxtamedullary nephron. So before moving on to the complexity of this topic, let's learn some major terms or jargons associated with this topic. So angiotensin is a peptide hormone that is part of the renin-angiotensin system that causes blood vessels to constrict and increase blood pressure. Then calcitriol is a hormonally active form of vitamin D. Then cortical and juxtamedullary nephrons, origin of cortical nephrons being in the superficial renal cortex, while that of juxtamedullary nephrons being near the renal medulla. Then erythropoiesis, the process of red blood cell production. Then erythropoietin, renal hormones that control erythropoiesis. Then nephron, basic structural and functional unit of the kidney. So these are the major terms associated with this topic. Next, multiple functions of the kidney. Let's start learning about this topic. Most people are familiar with one important function of the kidney, is to rid the body of waste materials that are either ingested or produced by metabolism. A second function that is especially crucial and critical is to control the volume and electrolyte composition of the body fluids. For water and virtually all electrolytes in the body, the balance between intake due to digestion or metabolic production and output due to excretion or metabolic consumption is maintained largely by the kidneys. This regulatory function of the kidneys maintains the stable internal environment necessary for the cells to perform their various activities. The kidneys perform their most important functions by filtering the plasma and removing substances from the filtrate at variable rates depending on the needs of the body. Ultimately, the kidneys clear unwanted substances from the filtrate and therefore from the blood by excreting them in the urine while returning substances that are needed back to the blood. Although this chapter and the next few chapters focus mainly on the control of renal excretion of water, electrolytes and metabolic waste products, the kidneys serve many important homeostatic functions including the following, like excretion of metabolic waste products and foreign chemicals, then regulation of water and electrolyte balances, regulation of body fluid osmolarity and electrolyte concentrations, then regulation of arterial pressure, regulation of acid-base balance, regulation of erythrocyte production, or regulation of erythrocyte production, secretion, metabolism and excretion of hormones, gluconeogenesis. So these are some other functions. Excretion of metabolic waste products, foreign chemicals, drugs and hormone metabolites. In detail, the kidneys are the primary means for eliminating waste products of metabolism that are no longer needed by the body. This product include urea from the metabolism of amino acids, creatinine from muscle creatine, and then uric acid from nucleic acids, and products of hemoglobin breakdown such as bilirubin and the metabolites of various hormones. These waste products must be eliminated from the body as readily as they are produced. The kidneys also eliminate most toxins and other foreign substances that are either produced by the body or ingested, such as pesticides, drugs and blood additives. Then regulation of water and electrolyte balance in detail. For maintenance of homeostasis, excretion of water and electrolytes must precisely match intake. If intake exceeds excretion, the amount of that substance in the body will increase. If intake is less than excretion, the amount of that substance in the body will decrease. Although temporary or cyclic imbalances of water and electrolyte may occur in various physiological and pathophysiological conditions associated with altered intake or renal excretion, the maintenance of life depends on a restoration of water and electrolyte balance. Intake of water and many electrolytes is governed mainly by a person's eating and drinking habits, requiring the kidneys to adjust their excretion rates to match the intake of various substances. Within 2 to 3 days after raising the sodium intake, renal excretion also increases to about a large amount so that a balance between intake and output is rapidly re-established. However, during the 2-3 days of renal adaptation to the high sodium intake, we are talking about sodium excretion and ingestion levels. So, during the 2-3 days of renal adaptation to the high sodium intake, there is a modest accumulation of sodium that raises extracellular fluid volume slightly and triggers hormonal changes and other compensatory responses that signal the kidneys to increase their sodium excretion. The capacity of the kidneys to alter sodium excretion as a response to changes in sodium intake is enormous. Experimental studies have shown that in many people, sodium intake can be increased to 1500 MEq per day, more than 10 times normal, to decreased to 10 MEq per day, less than 1/10 normal, with relatively small changes in extracellular fluid volume or plasma sodium concentration. This phenomenon is also true for water and for many other electrolytes such as chloride, potassium, calcium, hydrogen, magnesium, and phosphate ions. In the next few chapters, we'll discuss the specific mechanisms that permit the kidneys to perform this amazing feats of homeostasis. So, before moving on to the complexity of topic, let's look at a figure. So, this figure shows effect of increasing sodium intake tenfold from 30 to 300 MEq per day on urinary sodium excretion and extracellular fluid volume. The shaded areas represent the net sodium retention or the net sodium loss determined from the difference between sodium intake and sodium excretion. So we can see there is a rectangular shape and sodium intake and excretion are represented on the y-axis. Then we can say intake is there, sodium retention, sodium loss can be seen from that colored portion, and excretion is also shown. So extracellular fluid volume is also represented in the next figure, and according to the time or days at which this experiment is being conducted, so we can see there is a slight increase in the 0 to 8 days. After that, there will be a regulation. So next, regulation of arterial pressure. The kidneys play a dominant role in long-term regulation of arterial pressure by excreting variable amounts of sodium and water. The kidneys also contribute to short-term arterial pressure regulation by secreting hormones and vasoactive factors or substances like renin, and that led to the formation of vasoactive products like angiotensin II. Then regulation of acid-base balance. The kidneys contribute to acid-base regulation along with the lungs and body fluid buffers by excreting acids and by regulating the body fluid buffer stools. The kidneys are the only means of eliminating from body certain types of acids such as sulfuric acid and phosphoric acid generated by the metabolism of proteins. Regulation of erythrocyte production. The kidneys secrete erythropoietin, which stimulates the production of red blood cells by hematopoietic stem cells in the bone marrow. One important stimulus for erythropoietin secretion by kidney is hypoxia. Hypoxia in the sense oxygen deficiency. The kidneys normally account for almost all the erythropoietin secreted into the circulation. In people with severe kidney disease or who have had their kidneys removed and have been placed on hemodialysis, severe anemia develops as a result of decreased erythropoietin production. Regulation of 1,25 dihydroxy vitamin D3 production. The kidneys produce the active form of vitamin D, 1,2-dihydroxy vitamin D3 or calcitrol by hydroxylating this vitamin at the number 1 position. Calcitrol is essential for normal calcium deposition in bone and calcium reabsorption by the gastrointestinal tract. Then glucose synthesis is the next function. The kidneys synthesize glucose from amino acids and other precursors during prolonged fasting, a process referred to as gluconeogenesis. The kidney's capacity to add glucose to the blood during prolonged periods of fasting rivals that of the liver. With chronic kidney disease or acute failure of the kidney, these homeostatic functions are disrupted and severe abnormalities of body fluid volumes and composition rapidly occur. With complete renal failure, enough potassium, acids, fluids, and other substances accumulate in the body to cause death within a few days, unless clinical interventions such as hemodialysis are initiated to restore, at least partially, the body fluid and electrolyte balances. Then physiological anatomy of the kidneys, general organization of the kidneys and urinary tract. The two kidneys lie on the posterior wall of the abdomen, outside of the peritoneal cavity. Each kidney of the adult human weighs about 150 gram and is about the size of a clenched fist. The medial side of each kidney, the medial side of each kidney contains an indented region called the hilum, through which pass the renal artery and vein, lymphatics, nerve supply, and ureter, which carries the final urine from kidney to the bladder, where it is stored until the bladder is emptied. The kidney is surrounded by a tough fibrous capsule that protects its delicate inner structures. If the kidney is bisected from top to bottom, the two major regions that can be visualized are the outer cortex and the inner medulla regions. The medulla is divided into 8 to 10 cone-shaped masses of tissues called the renal pyramids. The base of each pyramid originates at the border between the cortex and medulla and terminates in the papilla, which projects into the space of the renal pelvis, a funnel-shaped continuation of the upper end of the ureter. The outer border of the pelvis is divided into open-ended pouches called major calyces that extend downward and divide into minor calyces, which collect urine from the tubules of each papilla. The walls of the calyces, pelvis, and ureter contain contractile elements that propel the urine towards the bladder, where urine is stored until it is emptied by micturition, as discussed later in this chapter. So let's look at a figure. So we can see kidney is there, ureter is there, bladder and urethra is there. So general organization of the kidney and the urinary system is given. We can see nephron, enlarged nephrons are shown. Then minor calyx can be seen. Then major calyx region, along with that papilla region. Then renal cortex is there, renal pelvis region, renal medulla, renal pyramid, capsule of kidney, then ureter. That initial region is called as the hilum, which through which passes the renal artery and vein, lymphatics, nerve supply, and ureter, which carries the final urine from the kidney to the bladder. Okay. Next, the nephron is the functional unit of the kidney. Each kidney in the human contains about 800,000 to 1 lakh nephrons, each capable of forming urine. The kidney cannot regenerate new nephrons. Therefore, with renal injury, disease, or normal aging, there is a gradual decrease in nephron number. After age 40, the number of functioning nephrons usually decreases about 10% every 10 years. Thus, at age 80, many people have 40% or fewer functioning neurons than they did at age 14. This loss is not like life-threatening because adaptive changes in the remaining nephrons allow them to excrete the proper amounts of water, electrolyte, and waste products. Each nephron contains a tuft of glomerular capillaries called the glomerulus, through which large amounts of fluids are filtered from the body, and a long tubule in which the filtered fluid is converted into urine on its way to the pelvis of the kidney. The glomerular apparatus contains a network of branching and anastomosing glomerular capillaries that, compared with other capillaries, have high hydrostatic pressure, about 60 mmHg. The glomerular capillaries are covered by epithelial cells, and the Total glomerulus is encased in Bowman capsule. Fluid filtered from the glomerular capillaries flows into Bowman capsule and then into the proximal tubule which lies in the cortex of the kidney. From the proximal tubule, fluid flows into the loop of Henle which dips into the renal medulla. Each loop consists of a descending and ascending limb. The walls of the descending limb and the lower end of the ascending limb are very thin and therefore are called as the thin segment of the loop of Henle. After the ascending limb of the loop returns partway back to the cortex, its wall becomes much thicker and it is referred to as the thick segment of the ascending limb. At the end of the thick ascending limb is a short segment and that has in its wall a plaque of specialized epithelial cells known as the macula densa. As discussed later, the macula densa plays an important role in controlling nephron function. Beyond the macula densa, fluid enters the distal tubule, which, like the proximal tubule, lies in the renal cortex. It is followed by the connecting tubule and the cortical connecting tubule, which led to the cortical connecting duct. The initial parts of 8 to 10 cortical connecting ducts join to form a single larger collecting duct that runs downward into the medulla and becomes the medullary collecting duct. The collecting ducts merge to form progressively larger ducts that eventually empty into the renal pelvis through the tips of the renal papillae. In each kidney, there are about 250 of the very large collecting ducts, each of which collects urine from about 4,000 nephrons. Next, regional differences in neuron structure, cortical and juxtamedullary nephrons. Although each nephron has all the components described earlier, there are still differences depending on how deep the nephron lies within the kidney mass. Those nephrons that have glomeruli located in the outer cortex are called cortical nephrons. They have short loops of Henle that penetrate only a short distance into the medulla. About 20 to 30% of the nephrons have glomeruli that lie deep in the renal cortex near the medulla and are called juxtamedullary nephrons. These nephrons have long loops of Henle that dip deeply into the medulla, in some cases all the way to the tips of the renal papillae. Thus, vascular structures supplying the juxtamedullary nephrons also differ from those supplying the cortical nephrons. For the cortical nephrons, the entire tubular system is surrounded by an extensive network of peritubular capillaries. For the juxtamedullary nephrons, long efferent arterioles extend from the glomeruli down into the outer medulla and then divide into specialized peritubular capillaries called vasa recta that extend downward into the medulla, lying side by side with the loop of Henle. Like the loops of Henle, the vasa recta returns towards the cortex and empty into the cortical vein. The specialized network of capillaries in the medulla plays an essential role in the formation of concentrated urine. Now, let's see some major points related to the comparison of cortical and juxtamedullary nephrons. Location: cortical nephron, cortex region, but juxtamedullary nephron, medulla cortex junction. Then proportion of total nephrons: 85% is cortical nephrons, but only 15% is juxtamedullary nephron. Then function of cortical nephron include filtration and reabsorption, but juxtamedullary nephron include filtration, reabsorption and creation of medullary or osmolar gradient. Then cortical nephron, length of loop of Henle is short, but that's very long in case of juxtamedullary nephron. Then ascending limb of loop is very thin segment in case of cortical nephron, but it's thin and thick segments are particularly seen in juxtamedullary nephron. Then vasa recta is absent in case of cortical nephron, but it is present and it's usually seen dipping into the medulla. Then peritubular capillary network is large in case of cortical nephron, but it's small in case of juxtamedullary nephron. Now let's see some pictures related to this. This is nothing else but the section of human kidney showing the major vessels that supply the blood flow to the kidney and schematic diagram of the microcirculation of each nephron that we will be discussing later. But this picture shows the section. So we can see renal artery is there, renal vein is there, segmental arteries are present, Then interlobular arteries and veins are there. Arcuate arteries and veins. Then interlobular arteries and veins. Next, when we look at this picture, we can see there are regions called afferent arteriole, arcuate artery, arcuate vein, then juxtaglomerular apparatus, efferent arteriole, glomerulus, Bowman capsule, proximal tubule, cortical collecting tubule, loop of Henle, distal tubule, peritubular capillaries, and a collecting duct. Then another picture, it shows the basic tubular segments of the nephron. The relative lengths of the different tubule segments are not drawn to scale, but an approximate information can be obtained from this picture. We can see descending limb, thin segment of ascending limb, loop of Henle, thick segment of ascending limb, then macula densa region, distal tubule, proximal tubule, cortex region, then connecting tubule region, Bowman capsule, cortical collecting tubule, then medulla region, medullary collecting tubule is there, then collecting duct can also be seen. Then another picture, schematic and representation of relations between blood vessels and tubular structures and differences between cortical and juxtamedullary nephrons. So in this picture we can see cortex region is there. So afferent arteriole, interlobular artery and vein is there. Cortical nephrons are situated. Juxtamedullary nephron is there. Then in case of medulla region, inner zone, outer zone, two separations are there. So in case of outer zone, there will be arcuate artery and vein, interlobular artery and vein, thick loop of Henle, vasa recta region. And we just look at the inner zone, we can see collecting duct, duct of Bellini, thin loop of Henle, and all those related structures. Thank you for listening.